wants to stay after class and learn more of the cell biology of MHC1 epitope presentation, I'll tell you. But if you don't care, then that's fine. Somebody needed the explanation to understand this slide a little bit more. Okay, so this is the one we left off on. Hey. So just a couple things. The questions help me to re-explain this. The MHC class 1 protein has 4 parts to it. 1, 2, 3, 4 parts. And this part is sticking in the plasma membrane. So the MHC class 1 protein is on the outside of the cell, right? But it initially had to be made on the inside of the cell. And you remember how you make proteins? DNA makes RNA, makes protein. When this MHC class 1 protein was being made inside your self cell, there was a self or a non-self, another protein being stuck in the protein binding cleft. And then that MHC1 protein with its self or non-self protein here was stuck in the plasma membrane. And now this is all outside the cell, so now cells of the immune system can come around and interrogate if there's a self or a non-self protein in that cleft. Okay. Let's see. That was MHC class 1. But guess what? There's an MHC class 2 as well. So this is a cartoon of MHC class 2. It's got 4 pieces to it as well. 1, 2, 3, 4 pieces of protein. But look, it's got 2 stalks that can connect to the plasma membrane. All cells of your body have class 1 MHC proteins. Only antigen presenting cells have class 2 MHC proteins. So if you are a cell in the body that has a nucleus, you will have an MHC class 1 protein in your plasma membrane. But if you are also an antigen presenting cell, you'll have an MHC class 2. And what are the antigen presenting cells that we've talked about?
The blood cells?
Pardon me? Dendritic cells are antigen presenting cells. Good. This gets really tricky. Another antigen presenting cell is a B cell. Not a T cell, but a B cell could be an antigen presenting cell. And then also, so dendritic cells, B cells, also macrophages. If somebody said macrophages, you're right. I remember I heard somebody say that. So here's an antigen presenting cell. I'll call it an APC. Here's its nucleus, where the DNA is. And it says, antigen presenting cells use receptors to recognize and engulf extracellular pathogens. So if you are a macrophage or a dendritic cell, what's the way we talked about that they can recognize a foreign cell? So if this let's say this antigen presenting cell is a macrophage. And here's bacteria out here. And it's got all its antigens and epitopes on it. How is this macrophage going to recognize that bacteria? PAMPs. Yeah. So the bacteria will have PAMPs, pathogen-associated molecular patterns. And the macrophage, what's the receptor that it will have? toll-like receptor. It'll have a TLR. So antigen presenting cells use receptors to recognize and engulf extracellular pathogens. So let's say that there's recognition occurred here. Now the macrophage has taken the bacteria inside. So the macrophage that is also an antigen presenting cell, it takes this guy inside and chops him up. So it takes the bacteria inside and chops it up, chops bacteria, and takes pieces of bacteria, those pieces are the antigen epitopes. Takes those pieces and then does what? Presents those pieces in MHC2. So we'll take those chopped up pieces of the bacteria, however you look, it'll take those pieces and put them in an MHC2. Let's see, how do I want to draw that? There we go, that's good enough. So it'll take those pieces of chopped up bacteria, put them in the MHC2, and then MHC2 will be stuck out here in the plasma membrane with the piece of bacteria, with the piece of bacteria in it. I'm the worst artist. What?
It's kind of morbid, isn't it?
What? It is. Well, Mother Nature is.
Process. Like you hang your enemies out in front of your house.
Yep. It's like chopping their heads off and putting them on the gates of Rome.
Yeah.
So everyone can
Like
see.
Games of Thrones.
Don't come near.
Don't come near. And don't.
This is what enemies look like.
Exactly. They have their heads chopped off and we present them to the world. Yeah.
It's kind of like, I don't know, like this might help other people remember. Like Sid would say, like MHC1 is what I made today.
Oh, yeah.
MHC2 is what I ate.
It's what I ate today.
Yeah.
I think I do have a cartoon down the road because another student told me that Sid said that. I thought that was a good way to
Yeah,
remember.
yeah.
Yeah, it's what I made today and it's what I ate today.
Mm-hmm.
So this is MHC2 is what I ate today because phagocytosis is eating. And then this is what I ate today, and then what I made today is I got infected and made something. Okay, yeah, I've got a cartoon for that. Yeah, you just fixing your hair?
Yep. Sorry.
Okay, no problem. So yeah, I wish I only got like blue and a crappy purple marker. I should have done it in different colors, but any questions? This is a lot of cell biology. It's really complicated. And it relies on the fact that you completely and totally understand protein synthesis from 201. And you remember protein synthesis. And then I build on that knowledge of protein synthesis, which you probably don't remember. So that's why it's difficult. But it's all in blue up here. Now once, then the story's not even over. The story gets even more complicated. So this macrophage that is also an antigen presenting cell presented the epitope in MHC2. Now another immune system cell can come along and recognize that. And then you're going to get a whole bunch more cell biology and biochemistry from that cell that's recognizing that complex. So then the next part of the story is another cell comes along and recognizes that and activates this immune system cell. So did you think you were going to take a break tonight after the test?
No.
You're like, no, I never thought that. And here's a cartoon of it that I just drew. So it doesn't show it in this cartoon, but the green thing is, they're just calling it an antigen, but it could be a bacteria with antigens on it, antigens epitope on it. And it doesn't show it, but how is this recognition between the macrophage antigen presenting cell and the bacteria? How's that taking place? Toll like receptors and PAMPs. Yeah, exactly.
Pam someone's kind of a adult diaper.
Adult diaper, yeah, Pampers, is that your kitty diaper?
I think Pampers are like baby diapers.
They're baby diapers, yeah. Well, yeah, so he recognizes the baby diapers on the bacteria, phagocytoses it, and then you do all this biochemistry and molecular biology, and then you present a fragment of that bacteria on an MHC2 protein, and then the other fragments of the bacteria, they just get voided out. They're not immunogenic. They can't make you sick.
Where do they go?
So
They get eaten by
Yeah.
like a lymphocyte or something?
Well, they were already eaten by the macrophage, and now this is just residue. Probably, it depends if it's amino acids in there, you can just reuse the amino acids. But it could be voided out in the feces or the urine. Yeah.
So is this MHC2 protein the reason why once you've been introduced to an infection or like an illness, you cannot get sick by it again unless it mutates?
No, that's going to be on an antibody that's an antibody production question. But you're right, that can happen. What time is it? When do we get out of here? Right now.
55.
Oh, no, you don't.
60 seconds, go.
50 or 55?
55.
All right. Fine, go. See if I care. See you Wednesday.
Whatever.